welcome to this episode of PharmTech's Pharma Fundamentals. In this episode, Robert Dream, Managing Director of HDR Company and member of PharmTech's Editorial Advisory Board, talks about the risks and regulations involved in hold times in biopharmaceuticals. We hope you enjoy the discussion.
I am Robert Dream. I worked for the industry for almost 40 years as a consultant, direct employee, and I worked in a few part of the industry worldwide, globally. In part one, introduction, we explored hold time in biopharmaceutical manufacturing, examining the key concept associated activities and the various complexities involved. We also compared biopharmaceutical hold times with those applicable to small molecule manufacturing to highlight their similarities and differences. In today's session, we will focus on the regulatory landscape governing hold times and examine the risk associated with their management and implementation. Regulations that govern hold times, manufacturers should be aware that there is no single universal regulatory limit for biopharmaceutical hold times. Instead, regulatory agencies expect companies to establish scientifically justified, validated, and documented hold times that ensure product quality, safety, and efficacy throughout the manufacturing process. Key regulations and guidelines include US FDA, CGMP Regulation 21 CFR Parts 210 and 211. Requires manufacturers to establish written procedures that prevent contamination and maintain product quality. Hold times for intermediate bulk drug substance and drug products should be supported by stability data and process validation. Landscape justification for hold times may result in regulatory observation during inspections. The ICH guidelines, ICH Q7, good manufacturing practice for active pharmaceutical ingredient, requires time limits for holding materials to be established where appropriate and supported by data. ICHQ8, pharmaceutical development, encourages development of scientifically justified manufacturing process, including acceptable holding conditions. ICHQ9, quality risk management, recommend using risk assessment to determine and justify hold times. ICHQ10, pharmaceutical quality system, emphasizes life system management, change control, and continued monitoring of manufacturing processes, including hold times management. So hold time management is a part of the process. It's not outside the process. European EU GMP requires manufacturers to define maximum storage periods for intermediate and bulk product. Hold times should be validated and supported by stability studies that demonstrate product quality maintained. PICS GMP guides aligns closely with EU GMP expectations and requires scientifically justified hold times appropriate storage conditions, and documented evidence supporting those limits. Process validation guidance. Regulatory agencies expect whole time studies to be incorporated into process validation or documented as separate studies where appropriate. Studies should evaluate the impact of storage durations and conditions on critical quality attributes, CQAs, including potency, purity, aggregation, bioburden, endotoxin levels, and stability. Best practices of compliance include establish maximum hold time for all critical process intermediates. Justify hold times with stability, microbial, and process performance data. Define and monitor storage conditions. For example, temperature, light exposure, container closure, etc. Reassess hold times following process changes or deviations. Maintain comprehensive documentation to support regulatory inspections and audits. Are there differences between regulations? Are there any different requirements for hold times? Let's look at a few items in here comparing FDA versus EU GMP. FDA regulations 21 CFR parts 210 and 211 do not prescribe specific maximum hold times. Instead, manufacturers must establish written procedures and demonstrate that holding periods do not adversely affect product quality, strength, purity, or safety. EU GMP provides more explicit expectations that maximum storage periods, hold time, for intermediate, bulk, products, and materials should be defined, documented, and supported by validation or stability data. Again, for US FDA, hold times justification is typically evaluated during inspection as part of process validation, stability programs, and CGMP compliance. On the EU side, inspections often expect predefined maximum hold times to be included in manufacturing documentation and validated as part of the quality system. Third item, US FDA guidance promotes a science and risk-based approach with hold times established using process understanding, stability studies, and quality risk management. EU guidance similarly support a science-based approach, but places greater emphasis on formally establishing and documenting maximum allowable storage periods. Common expectations in both regions, that is EU and the FDA. Regardless of the regulatory authority, manufacturers are expected to establish scientifically justified hold times based on stability and process data, validate or otherwise qualify hold times through appropriate studies, define and control storage conditions, for example, temperature, container, temperature exposure, etc. Monitor critical quality attributes, CQAs, including potency, purity, aggregation, microbial contamination, and degradation. Review and update hold times when manufacturer processes, equipment, or storage conditions change. Maintain documentation to demonstrate compliance during regulatory inspection. Practical implication for manufacturers. Defining documented maximum hold times for our critical process in intermediate and bulk materials. That basically, and it's an important thing. They have to define that. Supporting those limits with robust stability, microbiological, and process validation data. Using quality risk management principle, ICSQ-9, justify hold times and establish ongoing monitoring. So what are the validation requirements? US FDA does not specify fixed hold times limits for biopharmaceutical manufacturing. Instead, US FDA requires manufacturers to establish, validate, and document scientifically justified hold times that demonstrate intermediate bulk drug substance and drug product maintain their identity, strength, quality, purity, potency throughout the manufacturing process. FDA hold times validation requirement. Scientifically justified hold times. Manufacturer must establish maximum hold times based on scientific evidence rather than operational evidence. Hold times should be supported by stability studies, process development data, manufacturing experience, risk assessment, process validation studies. Hold time studies. Hold time validation studies should determine and demonstrate that material remain within predefined acceptable criteria throughout the proposed holding period. Studies typically evaluate drug substance intermediates, bulk drug substance, purified intermediates, bulk formulated product prior to filling, drug product before packaging, where applicable. The studies should represent outline manufacturing conditions, including maximum proposed hold duration, maximum and minimum storage temperatures, container closure systems, mixing or agitation conditions if applicable, product scale, equipment whenever possible. Critical quality attributes CQA testing. FDA expects manufacturers to demonstrate the hold times do not adversely affect product quality depending on the product testing may include potency, biological activity, purity and impurity profile, protein aggregation, fragmentation, charge variance, glycosylation profile where applicable, protein concentration, appearance, pH, osmolality, particulate matter, bioburden, endotoxin, sterility where applicable, residual host cell proteins, and residual DNA. Microbial control. Because many biopharmaceutical intermediates are aqueous solutions, FDA expects manufacturers to evaluate microbial risk during hold period by assessing bioburden growth, endotoxin formation, storage temperature effects, control strategy, frequency of monitoring. Integration into a process validation. Hold times studies are generally expected to be part of overall process validation strategy. FDA process validation lifecycle include stage 1, process design establish preliminary acceptance hold times. Stage 2, process qualification confirms hold times under commercial scale manufacturing conditions. Stage 3, continued process verification monitors process performance and confirms that established hold times remain appropriate throughout the product lifecycle and does not go out of bound. Documentation requirements. Manufacturers should maintain comprehensive documentation, including approved hold times protocol, scientific rationale for selected hold times, sampling plans, analytical methods, study result and statistical analysis, acceptance criteria, deviations and investigations, final conclusions and approval. Change control and periodic review. If changes occur that could affect product stability or quality, hold times should be assessed. Examples include process changes, equipment changes, container or packaging changes, manufacturing site changes, storage conditions change, scale up, or technology transfer. FDA inspection focus is on. During inspection, FDA investigators commonly evaluate whether manufacturers have scientifically justified maximum hold times, validated hold time studies representative of commercial manufacturing, demonstrated that CQA remain within specifications throughout the hold period, controlled storage conditions during hold periods, documented procedures and supporting data, appropriate change control, and periodic review procedures and processes. Risk that compromise quality and how to prevent them. During hold times, the period when a biopharmaceutical material is stored between manufacturing steps, material remain vulnerable to physical, chemical, microbial, and environmental risk. Risk that compromise quality during hold times and how to prevent them. Microbial contamination. Prevention. Use sterile closed storage containers, maintain aseptic handling procedures, control storage temperature, establish validated maximum hold times, perform routine environmental and microbial monitoring. Chemical degradation. Prevention. Store materials at validated temperatures, maintain appropriate pH conditions, minimize exposure to oxygen and light, use suitable stabilizing buffers, conduct stability studies to define acceptable hold times. Physical instability. Prevention. Avoid excessive mixing or agitation. Prevent repeated freeze-thaw cycles. Use validated storage conditions. Monitor product appropriate and critical quality attributes. Temperature excursions. Preventions. Continuously monitor storage temperatures. Use calibrated temperatures controlled equipment, intermediate alarm system for temperature deviations, document and investigate any exposure. Cross-contamination. Prevention. Use dedicated or properly cleaned equipment. Follow validated cleaning procedures. Segregate materials appropriately. train personnel in good manufacturing practice, GMP, container closure issues. Prevention. Use qualified compatible containers, verify container closure integrity, inspect containers before and after storage, select materials that have been validated for biopharmaceutical use. Next, we're going to look at what are some best practices. Best practices for design a whole time study. Use a risk-based approach. Simulate actual manufacturing conditions. Define maximum hold times. Monitor critical quality attributes, CQA. Assess microbial and microbiological risk. Evaluate environmental conditions. Use representative product batches. Conduct the study using commercial scale or representative pilot scale batches to ensure the result are applicable to routine manufacturing. Establish acceptable criteria before testing. Document and validate the study. Review and update product activity periodically. Third, what are the best practices for validating and documenting whole time studies and ensuring quality? Validation and documentation of whole times studies are essential. Excuse me. Validation and documentation of whole times studies are essential for demonstrating that biopharmaceutical material maintain their identity, strength, purity, quality, and safety during temporary storage between manufacturing steps. A well-validated and thorough documented whole times study provides scientific evidence that storage conditions do not negatively affect product quality and support compliance with good manufacturing practice and good expectations. Develop a comprehensive study protocol. Study objectives, materials to be evaluated, storage conditions, temperature, humidity, light, exposure, container types, etc. Sampling intervals, critical quality attributes to be tested, analytical methods, acceptance criteria, statistical analysis plan, representative of personnel. Responsible, it is of personnel. Use a risk-based validation strategy. Focus validation effort on manufacturing stage where material are most vulnerable to degradation or contamination. Consider risk such as microbial contamination, chemical degradation, protein aggregation, temperature excursion, cross-contamination, container containability. Simulate routine manufacturing conditions, production scale or representative batches, routine storage equipment, commercial container and closure standard operating procedures, normal environmental conditions. Use validated analytical method. All analytical methods used to evaluate product quality should be validated for accuracy, precision, specificity, sensitivity, and reproducibility. Typical quality tests include appearance, pH, potency, purity, protein concentration, aggregation, biological activity, sterility, bioburden, endotoxin levels. Establish scientifically justified acceptance criteria. Acceptance criteria should be defined before testing begins and based on product specified or product specifications, stability data, regulatory requirements, process capability. Maintain complete documentation. All aspects of the study should be thoroughly documented, including study protocol, batch information, storage conditions, sampling product, test result, instrument calibration records, deviations and investigation, statistical analysis, final validation reports. Investigate deviation promptly, perform periodic review and revalidation, ensure data integrity, integrate quality assurance oversight. Quality assurance should review and approve study protocols, validation reports, deviations, change controls, final hold time recommendations. Well, I hope you enjoy the two sections of this hold times review that what we went through, and thank you.